One of the biggest challenges we face, particularly for delivering chemotherapy drugs to children, is that we are limited by exposure of these, what is effectively toxic poisons, to healthy parts of the body. So typically chemotherapy is delivered through the bloodstream or taken orally, and the idea is that we're trying to get effective therapeutic concentrations to the brain, but we're limited by the doses we can give because of this widespread toxicity to the body. So because of this, one of the real opportunities we have is to deliver chemotherapy drugs what's called locally. So this is at close proximity to the site of the brain tumour. Quite often this is immediately after surgery, so we view surgery as a window of opportunity to deliver chemotherapy drugs close to the brain. And the second benefit to that is that it in principle avoids the systemic exposure to healthy parts of the child's body. Interstitial delivery is one of the very few drug delivery areas where there has been progress in recent decades. So from about the late 90s in America, there was the first of its kind interstitial drug delivery. It was a wafer that would contain one chemotherapy drug, and we looked at that as a rationale for basing our research on. So that was evidence of this is a drug delivery avenue that had some benefits. So the idea is that we particularly for malignant brain cancers, where we know the children and adults with these cancers, they almost always have surgery as the first treatment mode. So that's a window of opportunity for us to deliver something directly into the cavity left behind after removal of a tumour. So that's what the interstitial refers to, the directly in close proximity to the brain and where the tumour once was, and you've got a cavity left behind. The beginnings for this project was serendipitous, and we were quite fortunate. We didn't really have the expertise in these kind of fancy biodegradable polymers. It's just that the University of Nottingham, near our lab, we were exposed to Professor Kevin Shakespeare's pharmacy team, and they had developed this very fancy polymer in a very different application. It was made to regenerate bone, so it was to help mend broken bones. And we looked at it, and it was some certain properties of this polymer that our neurosurgical colleagues thought was particularly attractive for delivering chemotherapy to the brain. So in principle, the polymer is it's a powder, it's microparticles. But when you mix chemotherapy in a water solution, you can mix 1 or 2 chemotherapy drugs, it forms a paste, very much like the consistency of toothpaste or putty. And it's this intermediate property that is particularly attractive for brain cancer drug delivery. So the idea would be that immediately after surgery, when the bulk of that tumour has been removed, leaving a kind of irregular shaped cavity or hole behind, that the neurosurgeon would apply this paste to every nook and cranny into that irregular shaped cavity. And the next real neat thing about the polymer is that it only solidifies at the patient's body temperature within about 15 minutes. So by the time that the surgeon has applied this polymer, it will start to solidify and keep the shape to the cavity wall. And then in principle, the chemotherapy will gradually seep out over several weeks and hopefully will diffuse far enough to target those remaining cancer cells. We've done lots of laboratory and preclinical research just to demonstrate the clinical utility for this. So we've shown using several existing chemotherapy drugs that could be good candidates to deliver locally. We've shown that this polymer can gradually release these drugs over several weeks in the laboratory. We've also taken a sheep brain just to mimic the surgery that would be conducted in humans, and then to demonstrate the applicability of this chemotherapy containing paste. Then we took that sheep brain and we did various clinical scans that patients would get, such as MRI and CT, just to show that the polymer would not interfere with those scans, so that post-operation the clinicians could still identify any tumours, and this polymer that would be in their brains would not interfere. We also took the material and we gave the polymer a 6-week intensive course of radiotherapy, similar to what patients would receive, just to show that radiation would not disintegrate the material. So all these pieces of evidence indicate its clinical utility. And now we're at the stage of we are doing some work we've got mice that have got human brain tumours that are grown under the skin. So it's a very early test of the effectiveness of our polymer that's containing a few combinations of chemotherapy agents. Yeah, so I mean, one idea for this kind of a workshop is we're going to have an open discussion where I would like to invite my colleagues, where there's several drugs that may be very effective in the laboratory, just with when cancer cells grown in a dish are exposed to those drugs. But when those drugs are taken forward to an animal model or later on to a clinical trial, haven't worked, and they haven't worked either because of the high toxicity to healthy parts of the body, or because the drugs haven't entered the brain. So the brain is protected by what's called a blood-brain barrier, and this is an obstacle that often impedes the penetration of drugs. So for this kind of local delivery of interstitial delivery, we are particularly attractive to those drugs that could be really, really effective, but people have stopped using because it hasn't got into the brain. Those are the drugs that are really, really good candidates for us to consider for interstitial delivery. The initial stage of the research, we will use drugs that have been delivered systemically for brain cancer research. So our idea is that we will use the same drugs but deliver them better to see if it improves outcome. Beyond that, yes, this is a technology that should be widely applicable for future experimental agents that could be exciting. And it's something that only the kind of clinical trial toxicity data will help us gauge which drugs are safe to use and which combination and which doses. So our next step is to go into more clinically relevant animal models. So these would be mice that would have human brain tumours growing in the brain rather than growing under the skin. So one idea from this wonderful workshop that Children with Cancer have set up in London is to identify those collaborators, particularly in the United States where this research began, and to collaborate with them and work with our polymer but with their model. And then we can also not just treat the animals with the polymer, but also irradiate the animals and basically mimic what the human patients would receive. I think that the main reason why we are pursuing this as such an important area of research is that, particularly for the developing child's brain, systemic chemotherapy is often really, really devastating. So sometimes the treatments we offer to children may improve the survival, but it's to the cost of really significant morbidity and the quality of life. So I think ways to avoid chemotherapy, even in the first instance, is to deliver the same drugs, but this would be a way to deliver them much more safer and avoid that systemic toxicity.